Good day, Karandi and classmates. We are here in front to discuss about cell modification, including its types and example of each type. Our report is divided into three sections. First is we will be talking about cell modifications. Second, the types and examples of each types, including the functions of each types. And lastly, the summary about the discussion. Cell modification is hard to explain as it is in its basic form. You can already understand just by listening or reading it. Before anything else, we will now proceed immediately to the discussion. Let's start by breaking down the words cell modification. What is cell? Cell is the basic and fundamental unit of life. It is the smallest unit that can live on its own and that makes up all living organisms, including the tissues of the body. What about modification? Modification is the ability to modify a cell's genetic material in order to make it react to a particular stimulus. It is an alteration or change in a person, place, or thing that produces alterations. Cell modification is a technique used in order to modify cells, especially microorganisms, to be better suited for biological processes. It has been observed that some microorganisms are not able to grow or multiply well in a particular environment, and this can cause them to die out. Cell modification has the potential of altering the cell so that its abilities can be leveraged in the desired environment. The success of this is dependent primarily on cultural conditions where cells are grown and how they react with each other. Now, since we have understood the concept of cell modification, then let us proceed to its types. We have apical cell modification, basal cell modification, and lateral cell modification.
The apical cell modification. On all epithelia, they have one free surface called the apical surface, which is exposed body surface or at the lumen or space of the body cavity, duct, tube, or vessel. It is specialized to carry out functions that occur at these interfaces, including secretion, absorption, and movement of the luminal contents. Here are the types of apical cell modification. First, we have cilia. Cilia are small hair-like protuberances. Its main function is primarily for the locomotion, feeding, circulation, and aeration of the cells. In addition, they may be motile or non-motile. Next is flagella. Its characteristics include its unbranched, long, thread-like structures that are intricately embedded in the cell envelope. The main function of flagella is for bacterial motility. Another fact of flagella is they are accepted as being important virulence factor. Next, we have microvilli. Microvilli are characterized as membrane protuberances that arise from the epithelial cells. They are seen as small finger-like projections. It increases the cell surface area, which facilitates in the absorption of nutrients, ingested food, and water molecules. Lastly, we have pseudopods. Pseudopods, as its name means false feet, are temporary extensions of the cytoplasm. These cell modifications are responsible for locomotion and feeling. Here are the examples of each apical cell modification. First is cilia on the trachea. Cilia helps to remove contaminants from organs or tissue by helping to move fluid over the cell. These ciliated epithelial cells remove mucus, bacteria, and other debris from the lungs, beating in a synchronous rhythm to move these debris. Second is the flagella of the human sperm. Flagella allows the spermatozoa to actively reach and penetrate the female gamete, and they exhibit in a coiled motion. Third is the microvilli of the small intestine. The villi and microvilli increases the surface area of the small intestine, which aids in the absorption of bile salts and vitamin B12 through diffusion. And fourth is pseudopods on amoeba. Pseudopods serve two functions: locomotion and food capture. Amoebas use their pseudopods to ingest food by a method called phagocytosis.
Another type of cell modification is a basal cell modification. It is a process that eliminates the cell's greasy layer, creating a cleaner surface that's able to absorb things like makeup. It facilitates stable adhesion of basal cells to the basement membrane. Basal cells are found at the bottom of the epidermis, which is the outermost layer of skin. As new skin cells are produced, they push older cells towards the surface, where the old cells die and are slotted off. Basal cells are responsible for the production of new skin cells. When you have a cell phone, e-reader, or tablet with you, your body gets less sunlight and fewer vitamin D receptors. Basal cell production decreases, causing your skin to age, get sunspots, and wrinkles. A good example of B-cell modification can be seen in the hemidesmosomes, which helps any attachment or any cellular admission. The hemidesmosomes attach a cell to extracellular structures, such as the protein fibers in the basement membrane. The function of hemidesmosomes. Hemidesmosomes are multi-protein complexes that facilitates the stable adhesion of basal epithelial cells to the underlying basement membrane. The mechanical stability of hemidesmosomes relies on multiple interactions of a few protein complexes that forms a embedded tightly ordered complex. In other words, the reason why hemidesmosomes is an example of cell modification is that it provides strong attachments between these cells and other extracellular materials such as the basement membrane of the epithelial tissues. It means that a cell is modified to alter its underlying function into another functional cell.
Next is the lateral modification. It is a modification located on the sides of each cell. It is also called the cell junction because it is a specialized structure that serves as the intercellular connection between two adjacent cells. This helps the cells that come in close contact with one another to communicate and connect. There are three types of lateral modification: the tight junctions, adhering junctions, and gap junctions. First is the tight junctions. It is a junction that held two adjacent cells tightly. It forms a zipper-like protein barrier that cannot be easily crossed. This is to prevent the passage of unwanted materials across the cells. It is commonly found in epithelial tissue. An example is our skin and blood vessels. Next is the adhering junctions, or also called the anchoring junctions. It is a junction that fastens cells together but does not seal them tightly. It has an internal plaque attached to the cytoskeleton that is joined by intercellular filaments. It creates a sheet of cells that are strong but flexible. It forms organs where tissues must stretch, like the stomach and the bladder. And lastly, the gap junctions. It is a junction that connects cells with a protein channel directly from the cytoplasm, allowing a direct flow of molecules. This type of junction is important in hard and smooth muscles. Before we end, here's a summary of our report. Cell modification. It is a technique used in order to modify cells, especially microorganisms, to be better suited for biological processes. There are three types of cell modification. First is the apical cell modification. It is a type of cell whose main function is for movement or locomotion. There are four types, namely the cilia, flagella, microvilli, and pseudopods. Next is the basal cell modification. It is responsible for the production of new skin cells. Best example of this is the hemidesmosomes. And the last one is the lateral modification. It serves as the intercellular connection between two adjacent cells. It has three types: the tight junctions, adhering junctions, and gap junctions.